Can you guys see my slide move?
Yes.
Perfect. Okay, great. So just very quickly, my lab does a few things. Today, I'm going to tell you about some of the work that we do in the context of hematopoietic stem cell biology. And this is an interest that I have had since my PhD student years in Peggy's lab. My lab also studies the Hippo signaling pathway, which is a very important growth suppressive cascade, particularly in epithelial tissues. And its implications for cancer and malignancy. So I'm happy to talk to you offline about Hippo and all that good stuff. Okay. Perfect. So for this audience, we all know what hematopoiesis is, and my lab is very interested in trying to understand at a very granular detail how we actually go from a very rare subset of very early primitive cells to the number of mature cells that we have circulating in our blood and in our hematopoietic tissues. You guys are also familiar with this traditional view of hematopoiesis, which is sort of what we had whenever we started working on this. And I'll just highlight a couple of very important points here, as I think they will be relevant as we are testing some of novel ideas with some of the tools that we've generated. So in this pyramidal hierarchical view, which is what you guys would still find in a textbook if you open one on hematology, the idea is that at the top of this pyramid rest these very rare hematopoietic stem cells. Okay, and there are particular features of these cells that have been long to be, dogmas. Number one, the idea was that these stem cells were the main drivers of blood production, okay? Based on multiple pieces of data as well, it was thought that these were the only cells within this entire hierarchy that had the capacity to be long-lived and to have some capacity to self-renew over long term. And the general idea was that at least by nature, these hematopoietic stem cells would actually be multipotent, meaning giving rise to many, if not all of these lineages that I'm highlighting here, okay? In this, oops, I'm sorry, I don't know why that's moving automatically. I'll just go back. In this view, once hematopoietic stem cells divide and they do so asymmetrically in a steady state, then one of those daughter cells will become more differentiated progeny. And in this hierarchy, this cell is thought to be this multipotent progenitor cell, right? A cell that would have the capacity to give rise to multiple cell types, but that would be very transient, okay? And this mostly comes from, as I will tell you in a second, a collection of transplantation experiment over at least 2 decades, okay? Then these MPPs underwent further division and differentiation to give rise to oligoclonal and uniclonal unilineage progenitors. So this was the main idea that still is mostly ingrained in as far as how most people think about hematopoiesis. As I mentioned to you, that the majority of those concepts and the data that supported those graphs and those pyramids were mostly described from the experiment of transplantation. Bone marrow transplantation in this context, and just looking at the audience here, it seems that most of you guys are familiar with this, if not, you probably have done it in the lab. But this was obviously the main tool that people have had for decades, in which they took cells, be it a population of heterogeneous cells or very isolated and purified different cell types, and then transplanted them intravenously into animals that were lethally irradiated, that were myeloablated for the most part, okay? And what those cells produce, for how long they were produced, and how fast they were produced was sort of the main measure of the capacity and the biological features of the cells that you actually input into this system. But if you think about it very carefully, you can easily think that this particular experimental manipulation, oops, I'm sorry, I'm gonna have to turn off I think it's in automatic. Second, there you are. Oops. Perfect. So if you think about it, very carefully, right? So this situation does not really pertain to the hematopoiesis that is ongoing right now in my bone marrow, in your bone marrow, in the bone marrow of a mouse that is not being irradiated. And clearly, there are certain things that could potentially bias or I would say, exacerbate some biological behaviors in this context of transplantation. Right? Clearly, because of myeloablation, there is a massively damaged niche. There's niche cells that are dead. There's a cytokine storm. There's clear selective pressure and a strong selective pressure for these cells that you input to actually proliferate as much as possible. And so these, in our mind, could, instead of giving you a view of what's actually happening normally, could give you a view of what happens or what could happen in actually the worst case scenario. Okay, I'm seeing some activity in the chat. If there's something important, please let me know. Okay, got it. At the same time, you could argue that you could have cells that are normally perhaps long-term self-renewing and contributing at very high levels to hematopoiesis at a steady state, which might not be able to perhaps survive the transplantation process, right? So we thought that the data from these transplantation experiments could not so easily be extrapolated to what actually happens in steady state native hematopoiesis. So whenever I started my lab, I set up this idea this question to the people that came into my lab, to this question of how can we study blood production without doing transplants, without using this tool that had been around, for several years. And basically to answer the question of how is it that native hematopoiesis occurs, right? This non-transplant steady state hematopoiesis, and whether in fact the biology that we've discovered in terms of transplantation can extrapolate accurately to the native context. Okay. And before I start showing you some of the data, I just want to give you a sort of a big picture in terms of how we're thinking about some of these questions in the lab. And we think that it's very important for us to understand ancestry. And in the context of blood biology, for instance, we would like to understand where each of your stem cells come from, where each of those progenitors come from, right? Where your mature lymphoid B cells, whatever cells come from. Who is their sibling? Who is their grandpa? Right. So we think by understanding the source, I guess the cellular source, the ancestry, the lineage of these cells, we are going to be able to learn a bit more about their function. And sort of the second story that I will tell you about will make a point about that indeed the developmental source of the cells that you have, or the mouse has in some context, means quite a lot. Okay. The second thing that we think about is individuality, right? Not simply because cells look the same by a certain set of parameters, we should assume that those cells behave the exact same way, right? There's certainly a level of complexity underneath what we can see normally that needs to be considered, okay? So my lab, as I mentioned to you, to address this question of native hematopoiesis, has spent the last 8 years or so developing mouse and mammalian genetic tools to try to, as I mentioned to you, understand the function of stem cells, progenitor cells, and in general hematopoiesis at the single cell level in the native context, okay? And these, for the most part, allow for individual genetic labeling of cells and lineage tracing simultaneously of thousands of cells. And I'm highlighting some of the tools that we've generated. We published this inducible transposition barcoding model several years ago. The main story, or one of the stories that I will tell you about today, still uses this tool. We have now generated second generation barcoding tools that allow us to express the barcode so that it's also recognized using these novel single cell technology approaches. So you can get transcriptome and now epigenome measurements in addition to that lineage recorder that you have into your cells. And we can do this with lentiviruses, as done in these two papers that we've recently published. We also have now a mouse that is able to do this. So, and this is basically allows for inducible barcoding of whatever cell type that you're interested in, not only blood cells. And I think this is really going to open up new avenues in terms of really doing high resolution lineage tracing and developmental biology. We have also recently generated a set of reporter mice that allow the in vivo visualization using photo microscopy of stem cells, so that for the first time, we can actually directly visualize the behavior and interactions of these cells entirely in situ. And this is done in collaboration with Charles Lin at MGH. All right, so I will summarize a lot of this of the data that we've gathered in this one particular context of looking at stem cell contribution. Okay, so using this transposition tool that I will tell you a bit more about in a second. So this is just a conceptual view. The main surprise that we had whenever we were analyzing this data and lineage tracing thousands of clones in mice was that our data suggested that the long-term hematopoietic stem cells had a very limited contribution to adult blood production, right? Even if you were to follow these mice for up to 1 year, okay? Our data suggested that the bulk of blood production was not driven by these long-term stem cells, but instead by this population of multipotent progenitor cells, which again, in the context of a transplant, these cells only show long-term capacities that are, minimal, maybe 2 weeks to 4 to 6 weeks, depending on the population that you sort. And in our data, we certainly show that these MPPs could actually be self-renewing for at least 1 year in some of these animals. Okay. We had a follow-up paper here to sort of validate these results, again, using the Sleeping Beauty transposon system, but at the same time had another interesting observation, which argue that stem cells are indeed low contributors to most lineages, except for the megakaryocytic lineage, as we found a very strong coupling in terms of HSC lineage and megakaryocyte-only production. Okay, there's been a lot of work on this, so I want to spend my time telling you about this particular lineage. But again, I'm happy to talk to you a bit more about this data offline. So going back to that first result that I described to you, that stem cells are really limited in their steady state contribution to making blood. Right after we published our paper, Hans Reimer Rottewall and Thomas Hofer showed a similar thing using a traditional lineage tracing set of tools. But then there have been a different set of papers, including this by Boris Reises and this one by David Breiter that I'm showing you here, that suggested something a bit different. That suggested that indeed, if you do lineage tracing with some sort of CRE-R, HSC-enriched CRE-R systems, you actually were able to see somewhat significant contribution of stem cells into the periphery. And this has been a bit of a matter of debate in the field for a little bit, so I wanted to touch on this for a few minutes. So I've talked to Hans Reimer many times about this, and one of I think the critical points here is that whenever you do these lineage tracing experiments using a CRE-ER in this case, the specificity of the CRE driver strain is absolutely essential to make some of these conclusions. And this is, for instance, highlighted here. This is data from the Boris Reises manuscript. And in this particular animal, CRE-R, you can see that there is very selective labeling in the HSCs. But there's also some limited labeling in other populations, in other MPPs. And even though most of the labeling is in the HSCs, we have to understand that there's actually more MPP cells than HSCs overall in the bone marrow. So a 5% MPP might not be super clean, right? So in this case, you cannot distinguish what the source of this, assuming that this tomato label propagates, we cannot distinguish whether that label came from an HSC or a different progenitor cell. Okay, so we always thought that perhaps having a better labeling tool that would be completely specific would be able to provide some more insight into this still debated question. So I'll show you some data really off the press of a new model that we have that addresses this directly. okay? So can we trace stem cells using a more specific lineage tracing model, okay? You know, there's a lot of clonal data, but I think it would be, important for the field to be able to do this robustly with just a very, very straightforward Cree or a FLIPO-ER approach. And to do this, we have taken advantage of a dual genetic approach that we recently published in Nature. And this was the approach that we used to selectively label stem cells to visualize them via microscopy. But in this case, we're going to adapt this approach to perform lineage tracing. Okay. So this is a dual genetic strategy because it utilizes two different drivers, right? It's actually a 3-allele combination, but the idea is here. We have a reporter, at least in this paper, we have a reporter that is expressing stem cells selectively, but it's also expressing some MPPs. As I told you, right, we really want to be very specific and only label the stem cells. And to actually make this happen, what we do is we flux the reporter allele, like we did in this paper, and then we cross that strain with a FLT3 Cree allele. Okay, this FLT3 is a very useful allele because it has been shown to only be expressed as stem cells lose long-term self-renewal capacity. Okay, so if you have these mice, as you can see, even though normally you have GFP not only in the stem cells, but also in some potential daughter stem cells, short-term stem cells, MPPs, if that GFP is fluxed, then as soon as that stem cell will differentiate, then the FLT3 Cree will actually delete the GFP so that you would only have the reporter, in this case, selectively expressed in long-term hematopoietic stem cells. So we have adapted this approach, and this is the genetics for the ones of you that are interested. And in this case, we have the allele that we use to knock in reporters so that they are stem cell enriched is this EVI1 locus, MECOM locus. And this is one of the transcriptional start sites of that locus, MDS1. I can tell you more about that. But the allele that was knocked in was this H2B-GFP, which won't serve for much today, P2A, and we also knocked in a FLIPO-ER, okay? A tamoxifen-dependent FLIPO recombinase. And as I mentioned to you, this is completely fluxed. So if you cross that line with a FLT3 Cree, as mentioned to you, the GFP FLIPO-ER cassette will be very selective to the stem cells, right? It will not be expressed in any short-term stem cells or MPPs. And then obviously, we also add another allele that is in the ROSA26 locus that responds now to FLIPO recombination, right? So this is an FRT stop, FRT tomato. Okay, so in this line of mice, you could perform tamoxifen FLIPO-dependent recombination very selectively in the HSCs. Let me tell you what the data look like. This works incredibly well. So we were tremendously glad at how this worked. Okay, so this is looking at the SICKIT population of one of those mice given tamoxifen, just one dose, and then looking in the bone marrow for 48 hours. You see a very small population of tomato-positive cells, right, in the SICKIT enriched population. There's nothing in mature cells or any type of lineage committed progenitors. They're all SCA positive and SICKIT positive virtually. And as you can see here in this 48-150 plot, almost every single one of them is an actual long-term HSC. Okay. So now, again, these guys also carry the GFP to label those stem cells. And as you can see here in the SLAM gate, you have GFP positive cells and GFP negative cells. You can clearly see that we label about 50% of the GFP positive cells. Okay, so there is a fraction that is not labeled, probably none of tamoxifen getting into these guys. But the other thing that we have here is we have a GFP positive compartment and a GFP negative compartment, which we think are the sort of the immediate daughters of the GFP positive top HSCs, as you will consider them. Okay, so having this clean, robust, and very selective tool to label the stem cells, we wanted to perform long-term tracing, right? So basically here, we let the mice go into adulthood. We give them one dose of tamoxifen, and we either analyze the blood longitudinally. I'm not going to show you that. I'm going to show you bone marrow analysis at different time points after labeling to to see how and to what extent the label propagates from the very selective tomato in the stem cells to other hematopoietic compartments. So there's a lot of data here, but it's pretty simple. Okay, this is looking at percentage tomato. This is looking at the top stem cells, the bottom stem cells to keep it simple, and other compartments, short-term stem cells, other MPPs, and some more lineage-committed progenitors. Okay, so in general, we label about 20 to 30%, 35% of cells with one dose of tamoxifen. Obviously, at 1 week, this is in the marrow, we don't see anything. At 2 weeks, we see very, very little. And starting, I would say, between 8 to 12 weeks, you start seeing some label progression. Okay, and as we've shown repeatedly, and others, most of this label initially is in the megakaryocytic lineage. These are MKPs. We also have some platelet data that I'm not showing you. Okay, you can see that as time progresses, right, you start seeing now more contribution of the HSCs. But the point is that even after 1 year, right, if you look at the chimerism in your stem cells, it's about 27% or something like this with the data that we have. But if you look in most lineages, right, this label has not equilibrated. Okay, if you look, for instance, in megakaryocytes, this is about 8%. Okay, if you look in myeloid cells, this is about 5%. Okay, if you look in lymphoid cells, this is even lower, and that has been shown. So this, to me, adds to the body of work that suggests that a steady state, the contribution of the stem cells, I mean, there is some contribution, but it's very gradual, very slow, and in the overall scheme of things, limited. So here you could argue that less than 20%, about 25% of the granulocytes after 1 year of life have actually been produced by long-term hematopoietic stem cells. Okay. Using this model, we have started a bunch of experiments that test different challenges and see whether these stem cells can act as a reservoir for some type of emergency response. Okay, this is the same type of approach with PVS treated. We've treated with MCSF, as there's been some papers that suggested that MCSF can directly activate stem cells and allow them to give rise to myeloid cells. We see very little after 4 months of, this is a dual challenge of MCSF. Manu Pasek had a very elegant paper suggesting that IL-1 beta could actually induce proliferation and differentiation of the stem cells into the myeloid lineage. And as you can see here, again, just comparing the tomato labeling in different compartments and comparing to the control, you can see that there's probably some activation, right? But even, and this is a very chronic dose of about 1 month of administration, we see that there's actually limited contribution of the stem cells to different lineages. Okay, you have 0 even to the B and T cell fraction. So we're doing this more systematically. We're doing infections. We're doing poly-IC, and we have a positive control with semi-lethal irradiation that clearly shows that in those very, very major cases of hematopoietic loss, you do get HSCs into the periphery. But at least in this particular context, it seems that it is actually very difficult to acutely get stem cells, at least a bunch of stem cells, into the flow of a pathway producing mature cells. Okay. You can make a bunch of calculations and mathematical models, and I will spare you some of those. But compiling all of our data and using models that Hans Reimer and Thomas have developed, and sort of this is what they showed with their Ty2 ER mice. They argue that 0.9% of the stem cells actually differentiated per day. Our data suggests that it's even actually somewhat less. 0.2% of the stem cells go into this flow of differentiation. And this, again, this data contrasts drastically with the data from Boris's paper, suggesting that 3 to 8% of these stem cells go into cycle per day. So I do think that clearly the driver matters, the specificity of your driver matters, And we're very happy to have this now very robust tool in which we can try to test this in multiple contexts. Okay, and obviously we're doing some more clonal experiments to actually follow this up. Anyhow, it would be great to get feedback on this, as this is the first time that I actually present this data to anybody outside Boston.
Fernando, we have a question in the chat from Mark Perfetto, which is, what was the method of delivery for MCSF and IL-1B, and how frequent was the dosing in the mice?
Yeah. So these are following the protocols. The IL-1 beta was following the protocol by Manus Pasaget lab. I think they had a recent paper published in Nature Cell Bio, if not Cell Stem Cell. The details were exactly what we did in the paper, if I remember correctly. These are maybe sub-Q doses. There were several over a period of at least 3 weeks. MCSF, again, the concentrations, I don't know, but these were based on a science paper that was published several years ago. I think we did 2 challenges of MCSF, and I can give you more details if you want afterwards.
Thank you.
And if you have any sort of suggestions as far as what to use to actually mimic some sort of endogenous response, that would be fantastic.
Thank you.
Thanks.
Okay, following up on this particular observation, right, and assuming that this is really how stem cells are behaving, we wanted to address one particular observation here that is quite relevant. So in these lineage tracing experiments and in transposon experiments that we published several years ago, in addition to seeing the MPPs self-renewing, right, for many months, what we also observed was that there was a very limited sharing of clonal tags between HSCs and MPPs, even during a year of chase. Okay, and again, this was surprising because it has always been historically thought that these MPPs would be the immediate daughters, right, of these stem cells. So again, the observation that these MPPs did not share tags with the stem cells suggested that they came from the stem cells, maybe at an earlier time point in which we labeled, right? Maybe our experiments were using labels at 8 weeks of age. So maybe the HSCs gave rise to the MPPs before that time, right? Maybe they did it even before birth, okay? Maybe in the fetal liver. These are questions that still have not been addressed. And even a more sort of provocative population idea would be, perhaps, could these MPPs not, as thought, could come from a maybe developmentally distinct population and not necessarily from these HSCs, okay? So the experiments that I will share with you in the next few slides try to address exactly this question. Where do these long-lived, long-lasting MPPs come from? Do they come from the HSCs? And if so, when are they generated from HSCs? All right? So a very small primer on hematopoietic development, as I will talk about this, and I'm sure many of you guys are familiar with this, so I will just tell you the highlights here. Hematopoiesis occurs in multiple sites. There's a period at around 10.5 in which the definitive HSCs are specified, and many people have done amazing work on this, and this mostly occurs in this AGM region, and this is right before those stem cells are thought to migrate and see the fetal liver at around 12.5, with the main idea being that there's HSC expansion in the fetal liver, and that these stem cells now in the fetal liver then seed most of the blood system, okay? So that all of your hematopoietic system actually comes from the stem cells, from the definitive stem cells that were generated in the AGM, okay? And then, of course, the cells migrate into the marrow and then do their thing. Again, the paradigm is that these definitive stem cells establish most of your blood system at some particular time in development, okay? There's really very little data, hard data on this, except for this one lineage tracing from Hans Reimer-Rodewald that he published a few years ago, okay? So this is another question that we're gonna try to come to. In this, in the aorta around 10.5 or so, you as after you have the endothelial hematopoietic transition as these cells emerge, you guys may have heard of these clusters that form, right? These very early hematopoietic clusters that are a combination of cells that now, based on single cell data, are thought to be some more HSCs, some more progenitor-like, but nobody has really characterized them a bit functionally. So the data that I will show you would also speak to a bit about the identity of some of the cells in these particular clusters. All right, so where do these long-lasting MPPs come from? So, and how do we address this? I'm gonna show you two different tools to address this. The first one uses clonal tracing, okay? And this is using this transposon approach that allows for the inducible integration of transposons individually into cells, which then are used as genetic barcodes, okay, as we've published multiple times. And we thought that doing this in a completely in situ manner, particularly for the context of development, would be useful, as really most of the developmental hematopoiesis stories over the past 2 decades have involved the disruption of the embryo and transplantation, okay? And as Shannon McKinney has recently shown, if you do the experiments in situ, right, the answers in terms of how many stem cells we think there are early development change quite a bit, okay? So we'll do the lineage tracing using the transposon approach, but with inductions at different time points, right? Individual mice for different time points, so that we can actually go back, right, in terms of the of getting those lineages and trying to identify where in development does the HSC lineage diverge from the MPP lineage, okay? So it could be in the fetal liver, or it could be a bit earlier on, okay? So that's our goal with this clonal set of experiments, okay? So I've told you about the transposon system. It's been published, so I won't spend too much time telling you about this, except that it's inducible and that every cell carries a unique integration site that is random. And then we have ways of quantitatively detecting this transposon in sorted cell populations, which then allow you to make conclusions about sibling relationships and progenitor relationships, okay? All right, so the experiment is this. In some of these Sleeping Beauty mice, we give IV doxycycline. This is just one shot so that we limit the time window of transposition, which still is between, I would say, 36 hours or so. And this is done at multiple time points, right? 6.5, 7.5, 8.5, 9.5, and 13.5. We have a fetal liver time point. And then what we do is we simply let those mice make term, and then we actually let them go into adulthood, okay? Remembering that they were traced with one dose at different time points in development. So when these mice are either 3 months of age or a year of age, we can isolate the cells that underwent transposition and purify stem cells, MPPs, as we really wanted to figure out where this lineage divergence takes place, and also other mature lineage-committed cells, okay? All right, so this is a summary of 3 years worth of experiment by an amazing MD Ph.D. student in my lab, Sachin Patel, who's moving to UCSF in a few months. So I'll spend some time on this figure. The number here represents the time point of induction whenever doxycycline was given to these mice. This represents the different sorted populations that were analyzed and whose barcodes were looked at. And here, each row represents a unique barcode present, okay? And the color represents the frequency of that barcode in that particular population, okay? So in HSCs, and this is, the analysis is done in the bone marrow, that's important, in a 3-month-old bone marrow of mice that were induced at this particular time points, okay? So everything is analyzed at 3 months of age in adult. So in this particular guy, for instance, in this particular population, we found about 9 different barcodes, right? This one being the most abundant and so forth. If the same barcode is present in a different population, it will show up in the same row, okay? All right, so let's start thinking about this. if we induce at 6.5, which is basically a time point where the doxycycline would be labeling some sort of early mesodermal progenitor, what we see is that the barcodes are shared basically between every single population. Okay, again, suggesting that you probably labeled a mesodermal progenitor that will then give rise to HSCs and MPPs and everything. Okay, and this is quite early. This is useful because it also tells you how many of these progenitors were present at that particular time point in development. Okay, in this particular case, there was 9 what we think are mesodermal progenitors that give rise to the entire hematopoietic system in that mouse. Okay. All right, so what happens if you do the analysis if you do the induction at 7.5? You see something pretty much similar. Okay, you see more clones overall because at 7.5, you're probably labeling some sort of endothelial progenitor that is about to undergo a hematopoietic transition. Okay, so you can see most of the barcodes are shared. Okay, well, I'll leave this for the next observation here. Okay, so something interesting starts happening whenever you start labeling at about 9.5 and later. Okay. Again, these are HSCs. Sorry, the labels are not there. These are MPPs and the same populations across here. So what you find if you label a 9.5, right? And again, 9.5 doxycycline means that the mouse, the barcodes are being generated, I would say between 10 and 11, okay? Which is that moment when you actually have those clusters, okay? Generating some of those clusters and you actually have the largest clusters, about 10.5 or so. So what you can see clearly is that there are some clones here present in stem cells, present in MPPs, obviously, and that also present in the mature of hematopoietic progeny. But remarkably, you also see some clones present only in MPPs that are not present in HSCs. Okay, these clones are obviously giving rise to all types of mature cells, and you can clearly see that they are multi-lineage, right? Because they are crossed. We have some clones that are lymphoid only that I won't have time to tell you about today, but most of these are actually multi-lineage at the single cell level. Okay. This is even more striking if you label in the fetal liver stage. If you label at 13.5, again, time of true labeling 14 to 15, you can clearly see a small number of HSCs that are contributing, right? Again, when looked in the adult, right? But the majority of the stem cells, when are labeled in the fetal liver, seem to be mostly expanding, probably, right? Because they are not giving rise to mature cells. That job, again, just like what we saw in the adult, is actually left to MPPs, right? Multipotent progenitor cells that don't share a tag with HSCs, but still produce most, by 13.5, most, if not close to virtually all of it, of the hematopoietic and mature populations, okay? So what I'm showing you here is that you have this one population of cells that are non-stem cells that are able to be labeled very early in the embryo, starting at 9.5 or so, and that is still able to maintain production of multiple cell types in the adult mouse, okay? So we are calling these MPPs, EMPPs, embryonic multipotent progenitor cells, okay? And we think these are probably generated at around at the cluster stage, considering the time point of this labeling. It is certainly possible, right? And we can have more discussions about this, that there is a stem cell-like cell that would give rise to very early to MPPs, okay? But clearly that, if that was a stem cell, that stem cell was not retained as a stem cell, because otherwise we wouldn't see these MPPs without a lineage tracer in the stem cell population. Okay, told you about that. So our data suggests the existence of these long-lived embryonic progenitors, okay? And as I mentioned to you, do these EMPPs really develop independent of HSCs? And to try to address that, we will use an orthogonal tool to label these potential EMPPs. And so to do this, we made a knock-in mouse that, again, making use of this FLT3 locus, which is very unique because it's expressed only in the short-term non-transplantable stem cells. That's probably the best way to describe it. We knocked in a GFP, but also a CRE-ER cassette, okay? In an adult mouse with a CRE reporter, if you give 1 dose of tamoxifen, you can clearly see that you label most of the MPPs, you label most of the stem cells, and only a small fraction of the long-term stem cells, right? It's not perfect. You still see some. So that's important to know. Although if you change the reporter, instead of using a very efficient reporter as tomato, you can use something like YFP. We can get some mice that have about 30% MPPs and almost negligible labeling in the stem cells. Okay, so with this tool, we can specifically label MPPs. So we go into the embryo and we start in the fetal liver, and we give 1 dose of tamoxifen to these mice to basically answer the question: how long are these FLT3 positive embryonic cells? Okay, and so this is the data. Each line represents an individual animal, and you can clearly see that if you label MPPs, you see that most of your hematopoiesis is produced by these guys in the first sort of 6 months of life. Clearly, there's a decrease in the contribution, but I would say after 11 months or so, this contribution actually stabilizes in the myeloid compartment. So even after almost 2 years of life, when you label in the fetal liver, right, these are non-stem cells, they cannot be transplanted using traditional methods, you have lifelong self-renewing capacity of these cells, okay? So about, I would say 40%, 30% of your granulocytes, at least based on this model and this reporter, are derived from FLT3 positive fetal liver resident progenitors. The contribution is a bit biased towards lymphoid cells. If you look at T cells and B cells, for instance, which I'm going to show you, it's about 60%, right, of T cells that have this label, right, indicating that even during the entire lifespan of these mice, right, most of your T cells, at least in this graph, were actually produced by these embryonic non-transplantable progenitors and not by your traditional hematopoietic stem cells. We can also do this a bit earlier at 10.5. Again, about this around this AGM stage that I've told you about. And if you do this, the labeling is a bit less efficient for different reasons, but we still see long-lived, almost a year-long contribution into the myeloid lineage, which is about, on average, 15% or so. And you can also obviously see some lymphoid contribution from these FLT3 positive cells, which again is higher than the lymphoid in most cases. So we have here a FLT3 positive embryonic population that is long-lived and is lymphoid biased. This is just another quantitation of the lymphoid bias. I think this is important, right? So you could argue, well, Fernando, you still have 3% of labeling or 3 to 5 in the stem cells. How do I know that that label, that long-lived label does not come from that small number of stem cells? And that's clearly an important point. So if you take these mice, for instance, in this case, I think these are AGM labeled mice, and sacked at 9 months of age, okay, and you look at the tomato chimerism, in granulocytes, it's about 14%, for instance, in this particular guy. If you look in the MPP compartments, right, it reflects almost exactly what the label is in the granulocyte compartment, okay? The short MHC, it's a bit less, but the label in your stem cells is still very, very low, right? Suggesting that this 14% that you see after a year, 9 months of life, is likely not derived from these stem cells, which combined with our clonal transposon data suggests that there is a population here that is non-transplantable long-term that is actually giving rise to these granulocytes and obviously lymphoid cells for a very long time. We also went directly into the AGM, as that's the time point where we thought this divergence occurred between long-term stem cell population and a long-term non-transplantable EMPP population. And we tried to see if we could actually identify those FLT3 positive cells in situ. And this is 1 day after giving tamoxifen at 9.5. And what we clearly see is that there's cells that are parts of these clusters that are clearly FLT3 positively labeled, okay? Interestingly, we see these cells mostly in the larger clusters. When we focus on the smaller clusters, we actually don't see them, which suggests to us that something, as these clusters are increasing in size, probably cell division, there is some sort of interactions that actually allow for the specification of these FLT3 positive cells. We have done a lot of single-cell RNA-seq data that gives us more better molecular insight into this. Unfortunately, I don't have the time to talk to you about this, but I would love to do it offline. Okay, so I will conclude my presentation by telling you what we found. So we see, using two different methods, that there is an unexpected wave of HSC-independent progenitor-driven lifelong hematopoiesis contribution, okay? And we call these EMPPs, very basic. We believe that these EMPPs are likely specified in this cluster. So we don't think these EMPPs come from a different endothelial progenitor. And we have new data now to suggest that we think that there is a common progenitor, probably one of these pre-HSCs that these amazing developmental biologists have described, which then is able to give rise to either an EMPP type cell or a more sort of definitive HSC type cell. In the big picture, I think what I want to come across is that adult blood, in the mouse at least, is a mix of these two distinct lineages, an HSC lineage and an EMPP-derived lineage. And as I mentioned to you, we think most of the lymphoid production for life, in a mouse at least, does come from progenitors and not from stem cells, okay? And so if putting together all of our data, this is just a graph, not really data per se, we think that these dynamics are quite important, right? We think that, for instance, in the myeloid compartment, EMPPs produce most of your myeloid cells until midlife, and then it's mostly HSCs, which start contributing gradually, slowly. But in the lymphoid scenario, we think that even after 21 months of age, most of these cells are or have been produced by EMPPs, okay? So this, for us, would speak to potential implications of this data in terms of aging, right? As we clearly see that there's more myeloid production in aging. So one idea would be that HSCs are intrinsically, that the source of mature cells, in this case, HSCs, oops, where's my arrow? HSCs versus MPPs, would actually, could explain some of these phenotypes and this susceptibility to leukemia, which is mostly myeloid in the young and which is mostly lymphoid, myeloid in the old and lymphoid in the young. Okay, I will end with that. I had more to tell you, but I will end with that, and I will acknowledge my people. Sachin drove most of the work on the lineage tracing in the embryo data, so he's truly fantastic. And Sarah has done a lot of, Sarah and Alejo have done a lot of amazing work setting up some of these barcoding tools that I've told you about. Okay, and if you're looking for a postdoc position, please shoot me an email. We're actually hiring right now. Thank you so much.
Great, thank you very much. We have a few questions in the chat. Some of them relate back to the first part of your talk, so I'll start with those. One from Domingos Enrique, where you had quoted like a 0.2% potentially of the LTH HSCs. The question is, what does that number actually, that 0.2% actually represent as far as numbers?
Great question. So this is based on modeling, which, you know, I probably not the best at explaining. This is done with the same tools that Rodewald and Hofer have described. Francesca Mihor at the Dana-Farber helped us with the modeling here. But it basically means that if you put together all of the kinetics of the label and how it propagates, in this particular case, how it propagates from your long-term stem cell to your short-term stem cells over time, you can get this number that suggests that on average, 0.2% of the long-term HSCs are going into the pathway to make a short-term HSC, okay? You can make the similar calculations going from long-term to MPP and so forth, right? But based on this data, it would suggest that only 0.2% of the resident stem cells in the bone marrow will be going into this differentiation pathway and at least making a short-term HSC.
Okay, Domingos, do you want to ask more specifically about, you're asking about, like, is there a number that's associated with that? Let me just...
Yeah, I would like to know is the exact number if you calculate is 1 cell per day, 2 cells per day.
Oh, I see your point. Yes. So, I mean, in the mouse, depending on how you define your stem cells, I would say, I mean, let's give a number. There's probably 10,000 stem cells in long-term stem cells in a mouse, right? So, what's
200 a day.
Yeah, 1,000%. So that would be 1,100. That would be about 20 cells, I guess, right, per day.
Or I think it's 200. But anyway, it's a very, very, very small number. Yeah. Yeah, but I was thinking because a long time ago there was a physicist that did some calculations in how many really long-term hematopoietic stem cells exist in the bone marrow of a mouse, and he came to the number of 1, which means that it's so rare. In fact, that's what you see. What you see is that most is the MPPs, and in fact. You raise the question, is really there an hematopoietic stem cell, at least in these early things, or they have already give rise to the MPP, and these are then I would say transit amplifying population. So that compartment is the huge compartment in hematopoiesis. That's what you
exactly. No, totally. I totally agree with you. A lot of this is just nomenclature, right? So how do you actually define a stem cell? And I would argue, well, you can go both ways, right? But I personally would argue that some of these MPPs that we see that, you know, are there for at least a year of life and they give rise to more than one lineage could in theory be considered stem cells. They're just non-transplantable stem cells. As far as your question of whether there's one, so we've done some clonal tracing up to 2.5 years in some mice. And what you can clearly see is that after that period, the number of stem cells or whatever can be considered long-term contributing cells is reduced dramatically, right? So there's clearly a vast decrease in clonal diversity that we see. We don't get to 1, but there's clearly I would say almost a log-fold decrease in the number of stem cells that we see contributing in very, very old mice.
Thanks, great work.
Thank you.
Next question by Sergey Lulatov. It asks, these are beautiful data. Does the age of the mice matter when you're examining those same questions about the numbers? Like, do younger HSCs contribute more versus older HSCs?
Yeah, that's a fantastic question, Sergey. We are planning experiments in which we're going to induce in the adult, and I think that's going to look a bit different. Dave Breiter had some data in his paper with the FGD5 CREAR mice, in which he labeled in the young and the very old. And what he actually found was in the very old, there was a lot less contribution. Again, I don't know if it's because the label initially was more restricted to the stem cells in the old, which is what he shows in his paper, or there's actually clear biology. I think there's going to be significant changes. I don't know which direction, but just based on the clonal data that we have, all hematopoiesis looks very different.
Great, thank you. A comment from Janarla. Very nice talk. It's interesting that erythropoiesis reaches steady state at 8 weeks, it looks like. So I don't remember which slide that was on, but. So
maybe from the first part, I mean, it depends what you call steady state, right? So we're measuring the flow of label from the stem cell compartment to the other lineages. And we have the Weissman-defined MEPs here, which have, I would say, a similar kinetics, maybe a bit faster kinetics. Again, it's also slow than granulocytes, but clearly a lot faster than lymphoid cells, for instance, right? We also have some erythroblasts as well. So, if you're thinking about steady state, meaning that they're not longer contributed by the stem cells, I would say that you probably have to wait a bit more.
What I meant was really that if you look at the mouse developmentally, like you did from fetal liver, it goes to spleen and then goes to the bone marrow. By 8 weeks, all of the steady state erythropoiesis is only in the bone marrow. There's no any more erythropoiesis in the spleen or the liver, whereas the minute you bleed the mice, the spleen doubles in size. So the reality is what is happening in the dynamic, I really enjoy your dynamics of stem cells, but the same is true about erythroid lineage in terms of shifting from one place to the other during the aging of the mice.
Right. I get you now. So very, very good point. We haven't done, and we've been thinking about this, we haven't done very careful experiments looking at the spleen, as you're saying, that particularly during developmental hematopoiesis plays such a crucial, although transient role, as you mentioned. So we have experiments ongoing looking at, for instance, where the clones that go into the spleen, where do they come from, how long they last, and what exactly are making, and whether they are actually different than the clones as in the bone marrow in other places. That is a very good point.
I'll send you a paper we recently published where we followed quantitatively the exact number of erythroblasts in different places during aging.
No, I would love to see that if you send it to me.
Thanks, Mohan. Okay, a question from Hal Broxmeyer, which says multiple doses of MCSF and IL-1 beta to mice can have negative feedback on progenitors via releases of negative acting cytokines or chemokines. Could this be why over the time points that you analyzed, you're not seeing progenitor effects, mainly still HSCs?
Right. So let me see. This is our data here. So the goal of these experiments was to use these published protocols, which had suggested that stem cells go into cycle and differentiate based mostly on molecular data and on cell culture experiments. Okay, so if you look, for instance, at Manu's paper, when she gives IL-1 beta to mice, she sees stem cells going to cycle by BRDU, whatever. And whenever she gives IL-1 beta to stem cells in culture, they clearly produce more myeloid cells than the cells the HSC is not treated with IL-1 beta. Mm-hmm. So our goal here was to see if those surrogate measurements of cell cycle and or differentiation in vitro would also predict true stem cell differentiation into those particular lineages. For NCSF, we see there's really no difference, and this is 2 doses, no difference with the PBS control. For IL-1 beta, we do see some label progression, which likely means that some of the stem cells actually were pushed into giving rise to some mostly myeloid lineage cells, okay? This is still, I would say, very minimal compared to what Manu would have predicted from her data. Certainly, we cannot, and I cannot, and this was a very chronic challenge, right? I cannot rule out feedback loops that occur after this set of cytokine treatments. I imagine that's a bit complicated, but we're doing also clonal experiments with transcriptomic analysis to figure out exactly, for instance, in the case of the IL-1 beta, where exactly is this very small but significant contribution coming from? And is there some sort of signature that will predict that response?
I'm still a little worried because I know from our own work of years ago that when you give multiple injections of MCSF
okay,
or IL-1 beta, you actually decrease clonogenic progenitors as assayed by a colony assay. That's why I asked the question that maybe by looking at the time you're looking, you've already had a feedback, which is why you're not getting more of the progenitors.
Right. It's certainly possible, right? We have not looked at that. I mean, I guess we have to take a look at those papers. I mean, most of our, again, the way the target experiment was based on some of these 2 papers, but certainly I was not aware of this negative feedback on some of your progenitors, so we have to take a look at for sure. Thank you for bringing it up. Huh?
Yeah, then I had another question, if Diane could read it.
I'm happy to do that. Let me get back up to the questions. If I can get there, my cursor will go. Yes. So the second one is, even though you are using rigorous phenotypic analysis of stem and progenitor cells, it's clear from the work of many investigators that the phenotypes of these cells do not necessarily reflect their functional capacity. Do you have any thoughts or comments on that?
Yeah, no, absolutely, absolutely. That is clear, right? And we're very well aware of that. So obviously we use what is published that would enrich for, say, a stem cell versus an MPP population, but you certainly have this caveat. What these new barcoding technologies allow us to do is to, in addition to, again, perform tracing, you can do this in an unbiased fashion because you can simply analyze molecularly the cells. Okay, so instead of sorting, say, for CD48, EPCR, whatever, we can just put all of the KIT positive compartment, for instance, right, in the single cell RNA-seq machine, and it would tell us molecularly, right, what the cells are, and then we can assign lineage relationships. And whenever possible, we also try to do some functional analysis with populations that we have. Although, again, our goal is to try to limit transplantation.
Yeah, I still worry that you need to make sure that you have a functional capability to the cells that you're assessing.
Certainly, certainly. I mean, for us, the functional capability is, having a progenitor that is giving rise to multiple cell types over time.
Thank you. I really enjoy your work.
Thank you, Hap.
Okay, just a couple more comments. What's the implications of your model to the stem cell transplant? Should EMPPs be transplanted preferentially over the LT-HSCs, which is difficult to enrich?
Right. No, that's a fantastic question. So I would say that our data does not really negate any of the sort of transplantation data that has been collected in the mouse and the human for decades. It simply talks about what happens in a non-transplant setting. Now, the question is, what can we learn from our work that could aid, right, the transplantation context? And as you've alluded to, one possibility would be that since we know that these MPP or a fraction of these MPPs have long-lived capacity, but they cannot transplant, try to identify the mechanisms by which these MPPs are not able to sustain long-term self-renewal during a transplant, right? Is it an issue of conditioning, the niche? And is this something that we can actually pharmacologically or genetically rescue? Because if obviously we will be able to restore the capacity of these MPPs to be long-lived in a transplantation context, then I think we'll have a much larger pool of cells that could actually be engrafted long-term. And we're working on that.
Right, thank you. One final question, and then we'll just open it up for the last few people who are around. Thanks for the amazing talk. What's special about the clusters allowing EMPPs to arise? And do you think that it's intrinsic or extrinsic signal specific EMPPs? And then a final comment with that, sorry. Also, it's interesting that zebrafish EHTs don't form clusters.
Right. Yeah, the second one, right, it's very interesting. I don't know the answer. You have to lens on about that. But as far as your first question, I think that is a fantastic question. That's something we're really trying to understand. You put it exactly, whether it's cell intrinsic or cell intrinsic, right? Is this something completely random or not? So we are trying to address that question. It's a bit difficult, but again, we have some single cell RNA-seq data that is pointing us to the earliest changes in a cell that will take kind of FLT3 positive fate. And, we're also trying to do more granular epigenetic measurements, as I think probably there's some sort of priming, trying to identify the earliest moment of priming of these cells, which also will tell us a bit about sort of how they're actually generated. We're also trying to see if we can study this in vitro, obviously, as this would make it a bit easier to understand and to perturb genetically, but that is a very good question.
Thank you. Deanna, does that cover what you wanted? Not sure she's going to chime in.
Yes, thank you.
Okay, good. Well, thank you all for your attendance and participation. Really great talk. Really very thought-provoking, actually.
Thank you. You guys were awesome. So thank you so much.
Yeah, and thanks everybody for your attendance. And we hope to see you next month at the next seminar that's sponsored by the Centers of Excellence in Hematology. And thank you again, Fernando, for your presentation.
Thank you so much, guys. It was a lot of fun. Bye.
Okay. Bye-bye.
Thank you. Bye.
Thank you.